Welcome back. I'm gonna do a short recording on factors affecting skeletal muscle contraction. All right, so last time we had left off with after excitation contraction coupling, we had left off with a graphic that showed over time the tension developed in response to an individual skeletal muscle fiber action potential, right? So one action potential generates one twitch, right? And so those twitches can vary depending on, or vary with regard to their duration and their strength based upon the fiber type that we're talking about. So I'm going to expand a little bit from last time and show you a type 2X fiber type. That's the fast glycolytic. Let me label that. Say a little bit about metabolism later. There is an intermediate type that's also fast twitch. It would be the type 2A. And that's the fast oxidative glycolytic. Right? That's the fiber type that's intermediate. It's got both oxidative and glycolytic capacity, meaning it's decent at being both aerobic and anaerobic. The type 2X is glycolytic almost entirely during activity, which means that it's very good at being anaerobic. And the last fiber type, in red, is the really slow, weak one, right? Which is type 1, which is slow oxidative. That is the fiber type that, although it's slow and although it generates relatively little tension compared to the other two types, it is adapted for high endurance, right? Okay, so remember we had said that individual twitches really don't do much work because if we put a time scale on here, 100, maybe a little bit more than 100 milliseconds, or 1/10 of a second, and that's for the longest duration twitches. So not many things we do that quickly. So individual twitches, although they're the building blocks of overall muscle fiber tension and in a way, we'll get to whole muscle tension, individually they don't do much, right? Because they just don't generate enough force for a prolonged period. So what we need to have happen is we need to add these twitches together. Let's start over. And now, and I forgot to put it on the graph, the last graph here, but we would obviously have to have a stimulus arrive, an action potential arrive to get those twitches, right? So it's still tension, and it's still time. But now we could say seconds or minutes or whatever unit of time really you want. And so, but instead of having a single, and I'll draw one here, we'll do the fast twitch. I'm gonna shrink the scale a little bit so that I can show an increase in tension. They were too big on the previous one to go up from there. But so same size magnitude twitch, I've just changed my scale a little bit in tension. And so that's again, it doesn't matter which fiber type you pick because they all do this. But we're going to go with the type 2X. And if we get one action potential, we would get one twitch, right? Standalone twitch. However, if we increase our rate of motor neuron action potentials and therefore skeletal muscle fiber action potentials, I'm just going to have a bunch of arrows here for those on, right? Our twitches would occur, but then before the twitch could dissipate, its tension could dissipate, the next stimulus would arrive, which means that the intracellular calcium levels would rise over time during this repeated series of action potentials. And so as calcium increases intracellularly and we don't lose our tension in between, we get more and more cross bridges. And so our twitches sort of add together up up to some maximum up here, right, running out of room. And we call that addition of addition of twitch tension wave summation. And we could end up with some intermediate rate of action potentials where you would have some wave summation, but not reach full tetanus. You have to have a very high rate of action potential frequency to reach tetanus. Right, so again, once a motor unit is activated, it is doing one of three things. It's twitching repeatedly, or it's somewhere in the middle of wave summation, right? It generates some intermediate level of tension, which would then, be in here. We could draw that. If this was a little slower, you'd end up with something up, like this, where it would peak a little lower. Or complete tetanus, at the highest rates of action potential frequency, you get very high intracellular calcium levels, relatively, still higher outside the cell and in the sarcoplasmic reticulum, but much higher than they were originally, which means you get all the potential cross bridges being formed, and you get the maximum tension out of that. And so tetanus occurs for that reason, plus one other, right? So the first being a rise in intracellular calcium. The second being the fact that I'm going to draw my Hill model here. I'm going to draw it horizontally since I have more horizontal room here. And so here's a very simplified sarcomere representing the contractile elements of the muscle. Okay, so this is the Hill model. And we have, so this is the contractile elements, which is the sarcomeres, right? Those are thick filaments, those are thin filaments. This is what's called the series elastic element. We did mention the parallel elastic element, right? It's in parallel. Series means in line with, parallel means adjacent to, essentially. We don't really need to worry about the parallel elastic elements. Think of it as sort of a shock absorber. But when we get activation of the cross bridges, put some cross bridges here. Once cross bridges formation occurs, we would generate tension here and get shortening. But initially, some of that force goes into stretching the series elastic element, right? And so an individual twitch actually puts most of its force into stretching this, and not really much on the endpoints, right? In other words, we don't get a huge amount of tension on the tendons and bones with an individual twitch because it's just too fast and it's too weak. So if we shorten this for a prolonged period, wave summation or tetanus, we put a lot of stretch into the series elastic, making it more cable-like, right? Which means our force would be more effectively propagated to our tendons or bones, whatever you want to think of at the end here. And so that's another reason why tetanus generates a much stronger contraction, because this would be an extracellular recording of tension, not an intracellular recording of tension, just looking at this, right? Okay, so that's the usefulness of the Hill models. It shows us that we have to have prolonged contractions of the sarcomeres to stress the series elastic to really get significant tension on tendons and bones. Okay, so quick summary here is that this is one of the, it's a major one, but it's one of the three main mechanisms of increasing the tension in an individual muscle fiber, or you can think of them collectively in motor units, because every time we have an action potential down here, remember, it's not stimulating one fiber, it's stimulating all of the fibers within that motor unit, which is usually anywhere from a dozen to several hundred, right? So again, all of those fibers would be doing the same thing at the same time because they're receiving action potentials simultaneously. Okay, so that's the frequency-tension relationship, right? Wave summation and tetanus. The faster the frequency of stimulation, the more tension we get. The second main one, I'll do them in the order that I did them in lecture. Second mechanism of sort of altering or creating differences in muscle fiber or motor unit tension is differences in the cross-sectional area, right? So this would be the diameter tension relationship. So fibers that look like this in cross-section versus fibers that look like this in cross-section, right? This would be like a type 1, and this would be a type 2. Doesn't really matter which type 2. The type 2X would be bigger than the type 2A, but the relationship is still the same. Remember, our myofibrils would be running longitudinally through these fibers. The myofibrils are probably bigger in a type 2, plus there are probably more of them. And so cross-sectional area is proportional to the amount of tension you develop. So this fiber generates more force, right? This fiber generates more force than that fiber. Okay? Now, keep in mind that, well, let's back up. Work is the amount of force we generate multiplied by the distance over which we move something. So it's force times distance, right? So power really means, well, power really refers to bringing in the factor of speed. And so it's really how much work you can do, how quickly you can do a particular amount of work, right? Work is irregardless of time. You can mow your lawn in an hour, or you can mow your lawn in 4 hours. You do the same amount of work to do that. But if you do it in an hour instead of the 4 hours, you generated more power because you did more work per unit time. So power equals work per unit time. Right? So what that means is that although this one generates more force, this fiber type generates more force, it also generates more power because it is doing it more quickly. Right? And remember, the big difference between type 2 and type 1 fibers, the big molecular difference, is the myosin ATPase speed. It's a large determinant of power. I shouldn't say it's the only thing determining power, because also generating more force, right? Because it's all force is in work and work is in power, and so the more force would also generate more power. But the faster, so if we take two fibers that are similar in cross-sectional area, this, and they were the same fiber type, then they would generate the same force and the same power. However, if this is a type 1 and this is a type 2, right? So here force equals force, but power here is less than power over here, right? If this is a type 2 fiber, this is doing the same amount of work, generating the same amount of force, moving a load over distance more quickly, and so it has greater power. Okay? So that relates to, and that's important because doing work is important, but sometimes doing work at a particular speed is important, right? Getting up and leaving class, right, is important, but you don't want it to take an hour to get out of the room, right? You need to be able to do that amount of work over a reasonable amount of time. And more powerful fibers allow us to do work more quickly. Okay. The last factor, right? So now we have a diameter tension relationship. The last main factor affecting tension development in a fiber is the length tension relationship. So in this scenario, what we're doing is we're changing the starting length of the fiber, right? And can be done, this graph is actually done experimentally, where they take a muscle and stretch it to different degrees and then stimulate it. And so when the muscle is, muscle and muscle fibers are at low length, in other words, they are not dramatically stretched in the beginning, they generate relatively low tension. Right? You get a data point down here somewhere. But as we stretch it and then stimulate it, we start to get increases in the amount of tension that's developed. right? And so what we end up with is a curve that looks like this. And it will peak, sort of plateau, and then, experimentally at least, it would start to come back down, right? We'll make that dashed. And the reason we're making it dashed is that physiologically that doesn't really occur very frequently, right? So we're more concerned with sort of sliding up and down this particular scale with having our muscles start in a lengthened position versus muscles that are starting in a muscle fibers starting in a shorter position. And there's sort of a, more stretch is actually good in terms of generating, maximizing the amount of overlap of thick and thin filaments. And I've always done this in class, sort of where you've got your, your thick and your thin filaments. And too short is not good because overlap is not maximized. Stretched to optimal length is good because you have maximal overlap. And then overstretching starts to minimize overlap again. And that's an oversimplification, but it largely works. There are more details that go into that, but that's pretty much the gist of it. Remember, the zone of overlap, the whole point of the sarcomere is to generate the zone of overlap to generate tension, which can then be conveyed through the proteins of the sarcomere to the cytoskeleton, through dystrophin, through the costomeres at the membrane, and to the extracellular matrix fibers, right? The connective tissue layers surrounding the muscle. Because remember, we have to convert internal tension to external tension. It doesn't do you any good, right? As muscular dystrophies, right? It doesn't do you any good if you can generate tension inside the cells, but you can't convey it to structures outside the cell. All right, and so that's our length-tension relationship. Okay, I think I'll wrap it up there and do maybe a separate video on motor unit recruitment.